So now that our gel has finished running, we're going to transfer using a transfer membrane. So first thing we have to do is we're going to have to soak all our stuff that we're going to use ahead of time into the transfer buffer. So we're going to use these two sponges. We're going to make sure these are soaked inside our transfer buffer. This is to equilibrate them. And push down a bit. Make sure that they're soaked. We're going to let them soak and equilibrate. We also need to equilibrate a couple of filter papers. I'll just take a handful here. And we'll just start letting them soak inside. And then we're going to have to do our membrane. So first we're going to have to prime it with ethanol. So be careful. We're going to grab it from the corners here. Quickly into methanol. And then quickly into our soak to let it equilibrate into the SDS into the page. Now that we're done with the methanol, we can move the methanol aside. Now this is equilibrating. We can crack open our gel. So we're going to pull the gel that we have. We can take the top off. And we can start undoing our apparatus here. So we're going to do this. We're going to pour out. We don't need the liquid. We're just going to pour that out again. We're going to carefully undo our thing. These are the empty plates. We can move these aside. We won't need them. And this is our main plate. So we need to start prying these open. So preferably you start from the sides because we need to try to put these and try to separate them. So, all right, now that we've broken off the membrane, as you can see here, it's soaking inside our media, inside our transfer buffer really. And then after 5 minutes, it's been finished soaking, we can start working. So what we're going to do is we're going to take one of our cassettes, and these cassettes lock, so you're going to have to unlock these. We're going to work black down. And you can do the other way, but you just have to make sure the order is important. So we're going to take one of our sponges that's been equilibrated and soaked. And we're going to lay it on the black part here. We're going to grab a couple of our filter papers. Usually 2 should be fine. Kind of want to get the stack going equally here. Then you're going to want to grab your gel, and you want to be very careful not to rip it. So grab if you have tweezers, this would make it easier, but and you want to try and make sure that there's no gaps or air bubbles. If the top of the buffer is there, it's fine. So top of the gel. Then you're going to grab your blotting paper, your membrane here, and you want to overlay it pretty much over, as much as you can, over your gel here. You want to go as close to accurate as possible. You're going to grab the remainder. You grab 2 more. Cover it. And at this stage, what people usually do when you use best practice is you want to lightly go over this, pushing down. You're trying to get rid of as many air bubbles as possible. The air bubbles will impede on the transfer, so the less you have, the better. Now, some people are using rollers, and that's fine. Just whatever you have with you. Recenter this. Grab your last sponge and put it at the top. And this is where you want to close your cassette here. You're going to have to squish it until it can lock. And you want to lock it. Same thing as usual, black to black. Black to back here, and then there you go. Now we're going to get into, now we don't need the remainder of this. We're going to pour these back in. We're going to put our apparatus here. Remember, match black to black. We're also going to add ice packs here. This will heat when you do it, so we can kind of squeeze in here an ice pack to make sure it doesn't overheat while we're transferring here. And all that's left is for us to take this off with our buffer and top this off. The other side is filling. Make sure both sides are full. You want to go to blotting. This is not quite enough, but if we recycle the buffer here, that should be enough. And one more should be, just double check, and we're at the line, so we're good here. All that's left for now is to move everything out of the way. Now that we've cleaned up a bit, just to make sure there's no liquids mixed with the electricity here, is just for safety purposes. We're gonna cover this up here. Again, you should hear a click. Same thing as usual. Black to black, red to red. And this time we're gonna write it at 100 volts for an hour. And once you click run, you should be good to go. We're gonna click start. So this is a continuation of the membrane protein transfer. So we took out our membrane after the transfer finished and we put it into some blocking buffer. This buffer is just TBS along with some skim milk to help block in non-specific binding sites, and we left this overnight. Now we're going to start working on adding our primary antibodies, but before we do that we have to wash the membrane a couple of times to get rid of all the proteins and all the non-specific binding site proteins in the buffer. So the way we're going to do that is fairly simple. Holding down our membrane, we just want to pour the old buffer out. We want to make sure as much of the liquid is gone as possible. We're trying to remove this. And we're going to add some TBS. We want just enough to submerge it. We don't want to add too much, but just enough to wash it. If it's not moving in the shaker, that means you didn't add enough. So you just want add enough to suspend it. And you're going to want to have it shake. And you're going to wait 5 minutes between washes to make sure all the proteins are dissolved in the TBS. While we're waiting for our wash to finish, what we can do is we can start preparing our antibodies. So to prepare both the primary and secondary antibodies, we need to add 10 milliliters of TBST and 0.1 grams of BSA. We're going to prepare both of these. We're going to measure out 20 milliliters. We're also going to measure out 0.2 grams of BSA. We're just going to combine them. So you can do both of these because it's the same thing. All that will differ is just adding your antibodies right before you want to do it. So we're going to measure out 20 milliliters. Yep. Close that. I'm going to add your BSA crystals. I want to swirl it together. We'll make sure to dissolve the things on the side. This. Ideally, you do this in a beaker or other measuring apparatus. This will help ensure all the crystals are dissolved, but it shouldn't be too bad. And all you're going to do afterwards is allocate 10 mLs into each, and you'll have your primary and secondary antibody solutions ready. Okay, now that we're done all the wash steps, this is the final wash. We're going to prepare our primary antibody solution. So we've already prepared the 10 milliliters BSA plus TBST, and TBS, and now we're going to add our primary antibodies to it. So depending on the concentration of your antibody, you're going to add a specific amount. For us, it's going to be 5 microliters. So we're going to want to add 5 microliters of our antibodies. We have 2 different proteins, so we're going to add 2 different antibodies. So we're going to get ready to pull out our antibodies. Microliters, and you want to add this directly to your buffer here. I put a couple times up and down to make sure you've got it all. We can discard. And then now for our second primary antibody, 5 microliters. Because our primary antibody is not as concentrated, we're going to add a bit more of it for the actin protein, just to make sure we have enough primary antibody for the entire solution. And now that you've added your antibody, you want to make sure it's mixed throughout. So you want to kind of just roll it. You don't want to vortex it because you want to be too rough with it. You want to be gentle with it because they are susceptible to damage here, the proteins and degradation. If they're too rough with it, just mix it, make sure it's mixed throughout. Just rolling it and up and down. And set that aside for now. We're going to take our membrane off the shaker here and just dispose of the last wash like we've been doing. Kind of grab the edge of it. Make sure you guys get rid of as much of the liquid as possible without dumping your membrane. Now, simply just add your primary antibody solution. There you are, and just now you're gonna let your membrane sit in it for about 1 hour, and we'll come back after 1 hour and add our secondary antibody. Now that we've incubated our membrane and our primary antibody, we're gonna wash it a couple of times before we add our secondary antibodies. So it's pretty standard, like we've been doing. Right. Get as much of your primary antibody solution out, and we're going to wash it in TBS for the first wash. We're going to wash it at least 2 more times, 1 time with TBS, and then the final wash is going to be in TBST. So we're just going to add about 10 milliliters or so of TBS, and we're just going to wash. We're going to wait about 5 minutes. We're going to wait about 5 minutes for it to wash. Afterwards, we'll do it in a second and third wash step. Now that we've finished our third wash with TBST, we're going to start preparing our secondary antibody. And it's a similar process. We're just going to add our antibody to a solution we prepared earlier. So you're going to grab your antibodies. Depending on the concentration, you're going to add the required amount. In this case, for one of the antibodies, we only need 1 microliter. So we're going to grab our pipetter. Sure everything is open. Set to 1 microliter. Grab the required amount of antibody and add it to your solution. I pipe it a couple times up and down just to make sure all of it is released. You can close that off. Now it's time for our other antibody for our other protein. This one requires 5 microliters, so same as the primary antibody. And same process, you want to put it into your solution and pipette a couple times up and down just to make sure it's added. Now that both antibodies are added, we just have to mix our solution up gently. So we can roll it a couple times up and down. Now that that's ready to go, we can remove our membrane from our TBST. Remove as much as you can without having the membrane spill. Then you're going to add your solution that we prepared with the secondary antibodies inside. You can discard this. And we're going to put this back on the shaker, and we're going to let it shake for another 30 minutes, incubating our secondary antibodies, and we'll come back when it's done. So we've washed the secondary antibody off camera, similar steps to using TBST. Now that we're done the third wash, we're ready to add our substrate to help visualize our proteins. The way we're going to do this is we're going to alicate 9 mL of deionized water. Then we're going to add 1 mL of Opt4CN diluent. And we're going to add 200 microliters of the substrate for Opt4CN. And it's going to help us visualize our bands with our eyes, and then when we take it to the ChemiDoc, we're going to be able to visualize our bands better from that point on. So first we're going to do is we're going to prepare it. So I've alkylated these into smaller tubes to make it easier. So we're just going to alicate 1 mL of the diluent. That's 1,000 microliters. Have that set, ready to go. So we're just going to take pretty much all of it. We're just going to add that to our substrate liquids. Close this off. We're going to take 200 microliters. We're also going to add that over here. This is our substrate. Now we're just going to mix to make sure everything is homogenized. And we're going to add that and wash our membrane with it. So just simple ups and downs. Same way we've been doing it. We can close this. Push these to the side. So we have roughly 10.2 mLs in this Falcon tube. Make sure it's thoroughly mixed, and then we're going to wash our membrane in it. So we're just going to set this to the side. We're going to take our membrane, and we're going to dispose of the wash. Same way we've been doing with the last wash step. So ideally, we get as much of the liquid as possible out without dumping our membrane. We're just going to add our wash here. Again, never over the membrane, always to the side. sure you get all of it. Cover it. And we're going to put this on the shaker. And this can sit on the shaker for 15 to 45 minutes. We should be able to start seeing our bands visually. And we'll especially see them under special lights in the chemi dock. Okay, so now that we have let our membrane sit in our solution for 15 to 45 minutes, we can start seeing some bands. We're going to image it under the chemi dock to see the bands more clearly and as well save the picture. So what we're going to do is we're going to remove it from the solution. So this is, we're going to be careful not to dump our membrane here, but we can kind of just dump the solution into the waste. Then very carefully from the corner, we're going to grab the membrane and lay it onto your imaging dock here. This has already been pre-cleaned and you're going to have to clean it afterwards as well. And now we're going to take it to the dock. Hello everyone, we'll be talking about how to image your Western blot membrane after transferring onto your membrane. So first we're going to go to your imager, the chemi dock. You're going to want to turn that on over here. It's going to take a while to warm up and turn on, and once it's on, you'll get to this screen over here. You're going to want to click, you're going to want to sign in using the MBB lab, and you'll get to a screen that's something like this. Now, there's different options and functionalities, but we'll be talking about how to take the membrane images and how to measure your Western blot bands. So first, you're going to make sure that this dock is the appropriate one. You're going to want to use the BUV stain-free tray. It's a black tray. There's a white one and a blue one, but the black one is used for imaging. If it's the wrong tray, you can easily remove it and put it back. You'll hear a snapping once it's appropriate, and then you'll see that it'll confirm onto the screen. So you're going to want to push that back in while you're not using it until you've decided what type of imaging you want to do. Then you're going to want to select your application and what type of imaging you want to do. So if you're doing DNA, you'll click nucleic acid. If you're doing Kumasi, you'll click Kumasi blue. But in this case, we're doing blots. You're going to click blot. And then you're going to select chemiluminescence, depending on what you want. If you're using stain-free, you'll select stain-free. But for this demonstration, we'll be selecting chemiluminescence. And afterwards, the second thing you want to play with is your exposure time. You have the option to do a manual exposure time in case you know exactly how many seconds you want it to be exposed for, or you can do auto exposure, and this is the most recommended one. You have optimal, which will make your band quality better, or rapid if you're short on time. I always recommend you use optimal unless you're short on time or have any other reasons or know what you're doing specifically. Once you have your settings selected, it's time to put our gel in, or our membrane in this case. So you're going to take the tray out, and you're going to make sure it's clean, clean enough. Use water and Kim wipes if it's not before you use it. Then you're going to put your membrane, you're going to put it down over here. And follow your protocol if there's any chemical application you need to do on the membrane beforehand. After everything is ready to go, you're going to push it in until it can't go any further. You're going to close the tray. Now you should be able to see some sort of image. It might not show bands yet, but that's all right. After you're done and everything's good to go, reconfirm your settings. Chemiluminescent blot in this case, or stain-free depending on the protocol. Auto optimal exposure. And when you're good to go, you're going to click the bottom button over here and click start. So it's going to start warming up here for imaging. And then it's going to start setting the exposure time. So it's going to try to optimize it. So you'll see it going cycling until it finds the optimal exposure time. It will expose your membrane towards it. So you're just going to have to wait patiently until it does. In this case, it decided 17.825 seconds was the optimal. And we'll keep doing so until it finds the best quality for your image. This differs from number of antibodies, antibody concentration, protein concentration. So it's best to let the imager decide what's best. And once you've done, this is what we've seen results. So this is touch screen, so you're also able to zoom in and able to play with the settings. And as we can see here, there's clear quantification of our bands, from increasing concentration to decreasing concentration of proteins. You can see different lanes and different bands, and you can see different bands and different lanes indicating different proteins of different sizes. From here, you can take your image, you can resize it, and when you're ready to go, you can click send, and you can export it to a USB for further analysis. This is how you're able to image your membranes and I hope it's easy. Now that you have your membrane, it will be auto-saved over here. So if you ever want to come back to your membrane, you can click Gallery, and you're able to see all the images that have been taken on this machine on a specific date. So you can always come back to this on a later date and collect your sample. Once you're done, it's important to make sure everything is clean and ready to go. So you're going to open up your tray, turn back to the camera setting, and remove your tray gently, and remove your membrane from the sides. This is a test membrane. But then you also want to make sure that you're able to clean everything. So using water is important. So add just a little bit of water. This is a bit too much. Then you're going to want to grab Kim wipes and just kind of wipe it. Be careful not to use actual wipes, as they may scratch the surface and may cause poor imaging quality later down the line for others as well. So once you're done, make sure it's not wet before you put it in. So if you use another Kim wipe, feel free to do so. You want to make sure it's not wet before you put it in, as it could cause problems. So you want to make sure any chemicals that you may have used or any buffer doesn't remain here, as this may lead to scratching and may lead to degradation of the membrane and of the imager as well. It's important to take the extra seconds in being thorough in your cleaning rather than risk damaging the expensive equipment. So multiple Kim wipes are needed, and your water will slowly evaporate as well. So now that it's mostly dry, you're good to go. You just return it in, and always make sure the tray is closed. If you're the last one using the machine, sign out using the log out button on the top. And if you're at the end of the day, click this button, you can turn it off, and it'll be ready for the next day to use.